This is Dr. Padala Tirupati, Assistant Professor, Department of Botany. మనం ప్రీవియస్ లో జరిగినటువంటి పీజీ ఎంట్రన్స్ కి సంబంధించి అంటే కామన్ ఎంట్రన్స్ టెస్ట్ సెంట్రల్ యూనివర్సిటీకి సంబంధించినటువంటి ప్లాంట్ సైన్సెస్ బాటనీకి సంబంధించి ప్లాంట్ సైన్సెస్ కి సంబంధించినటువంటి ప్రీవియస్ పేపర్ ను మనం ఈ రోజు విశ్లేషణ చేసుకుంటున్నాం. దానికి సంబంధించి క్వశ్చన్స్ ను మనం ఇంగ్లీష్ లోనే చూద్దాం, ఎందుకంటే టోటల్ గా ఇంగ్లీష్ లోనే క్వశ్చన్ పేపర్ ఇవ్వడం జరుగుతుంది కాబట్టి మై డియర్ ఫ్రెండ్స్ మనం చూద్దాం ఒక్కొక్కటి. So part 1 contain 25 questions and part 2 contain 75 questions. Total 100 questions are here. Okay, my dear friends. So మనం దీనికి సంబంధించి మనం చూసేటువంటి క్రమంలో right. So one of the following is an instrument used to measure water potential. So water potential is measured by, water potential of leaf tissue is measured by psychrometer. Psychrometer is a measuring instrument for the water potential, measuring of the water potential. The water potential term has coined by, given by Slater and Taylor. Okay. Then the organism that is suitable for mapping the gene with respect to centromere is, so these which organism is suitable? So that is Neurospora crassa. It is a blue mold or green mold of Ascomycotin of fungi. Then the strain is used to test seed viability. So the ability to germinate of the seed, that is we can call as seed viability. So seed viability based on the pink color developed by the living seed tissue contains one of the following salts. So which salt is useful for the testing, for the using of the pink color for the seed viability test? So tetrazolium salt is used. Okay, tetrazolium. Then fourth question, a cross was made in snapdragon. Okay, snapdragon plant between AB, capital AB and small ab, cross with small ab, small ab. What portion of the progeny will be small a, small b, small b if the two genes capital A and capital B are 24 map units apart? So the right answer is 38%. The fifth question, the key switches in signal transduction circuits regulating gene expression are transcription factor, removal of male organs from a hermaphrodite. Hermaphrodite flower is nothing but bisexual flower. So hermaphrodite flower, to prevent selfing, is known as emasculation, removal of the stamens. So male organ means stamens, so that is we can call as emasculation in hybridization experiments. Okay, then the seventh one, the mating of plants in all possible combination is known as diallel mating. The arrangement of petals and sepals in a flower bud before it opens is known as estivation. So estivation, we know that valvate estivation, imbricate estivation, twisted estivation. So this type of estivations we are studied in this arrangements of the petals and sepals before opening the flower. Okay, then when a cell is fully turgid, which of the following will be zero? So that is a suction pressure. Hydatodes are water secreting glands. Okay, hydatodes are water secreting glands which is contain epithelium tissue. Epithelium, it is a loosely arranged tissue, epithelium tissue, which is present in hydatodes. So that is water secreting glands. This is a special type of tissue in especially herbaceous plants like tomato and other grass plants. And deamination of 5-methyl cytosine produces thiamine. Okay, then once centimorgan is defined as the genetic distance between two loci with a statistically corrected recombination frequency of 1%. 1% is the right answer. Which of the following amino acid in proteins cannot be post-translationally modified? Alanine. reverse transcriptase is. So reverse transcriptase, which is a special type of enzyme present in RNA genetic material organisms like HIV, polio, and corona, SARS-CoV-19, COVID-19 virus. So this reverse transcriptase is RNA dependent DNA polymerase. So if it is a transcriptase, that is DNA dependent RNA polymerase. If it is a only transcriptase, that is DNA dependent RNA polymerase. But the question is reverse transcriptase, that is RNA dependent polymerase is the right answer. Okay. Then a plant species nearing its extinction due to virus infection has been given for tissue culture and micropropagations. Scientists should choose the below 4 explants for culturing. So in this 4 explants are here: meristem, shoot tip, leaf tip, root tip. Remember, my dear friends, so pathogen-free plants, especially virus-free plants, produced by the using of the meristem tissue and shoot tip tissue. So the right answer is 1 and 2. The right answer is 1 and 2. Meristem, shoot meristem and shoot tip also. Then the next question, consider the following statements on the common features between phase contrast and dark field microscope. So this question comes from microscopy. So the right answer is 1 only. So that means both increase contrast between specimen and background without staining. So without staining, which is increase the contrast between specimen and background by the using of this phase contrast dark field microscopy. Okay. Then how would you measure the transcript abundance of a particular gene? So that is by performing northern blots. So the northern blot test is used for the identification of the RNA. So the transcription means DNA dependent RNA polymerase. In the presence of DNA dependent RNA polymerase, that is nothing but transcriptase enzyme. In the presence of the transcriptase enzyme, from DNA to produce mRNA, that we can call a transcription. So mRNA is synthesized or not by the using of this northern blot test. If we can use southern blot test, that is identification of the DNA. If we can use western blot test, that is identification of the protein test. Okay, protein. Then, though DNA and RNA are nucleic acids, isolation of RNA in the laboratory requires extreme precautions and pre-preparations than isolating DNA. This could be because RNA is unstable and protein and prone to degradation, but DNA is relatively stable. That is DNA is isolation. DNA isolation. So this is the right answer. Then identify the mismatch. See here. So zooxanthellae, that is a pyrophyta. It is correctly matched. And Cephalurus, it is a parasite. Cephalurus is one and only parasite in algae. It is caused red rust disease in tea plants. Okay. Then Eulothrix. Eulothrix is a branched filaments is wrong match or mismatch. It is the wrong answer. Red algae, we know that picoerythrin, which is one of the which is one of the phycobilin protein, phycobilin pigments, which is present in rhodophysi members, algae members. It is also correct. So the question, what is the mismatch of these options? That is Eulothrix. It is not a branched filament. It is a filament, but not branched filament. Then, so match. See here, waxy cuticle. Waxy cuticle is that is a restrict water loss. Okay, and a thick or lignified cell wall. That is mechanical support. It is useful for the mechanical support, additional support to the plants and cell. And homeohydry. That is, we can call as vascular system. Vascular system, xylem and phloem. and pigmentation, that is protection against excess light. Excess light. So the correct option is B. Then plankton will be absent in lentic biome, marine biome, lakes. So these are all planktons, which is free floating organism of the plants on the surface of the water. So that's why lentic is a stable water, marine is a stable, lakes is also a stable, but lotic biome, it is a flow, it is a flowing water. So flowing water unable to present in these planktons. So that's why A is the right answer, lotic biome. Then the most commonly used stain for producing bands on chromosomes for karyotyping is, that is GIMSA. GIMSA, okay, GIMSA. And cistern is the genetic unit of biochemical function. Cistern is biochemical function, genetic unit of biochemical function. It is nothing but DNA. Okay, so if it is a recon, the smallest unit of recombination. Okay, if it is a mutant, the smallest unit of the mutation. Okay, mutant. So according to Seymour Benger, so gene is differentiated into 3 units. One is cistern, recon, mutant. Cistern is a genetic unit of the biochemical function, and mutant is the smallest unit can undergo the mutation, and the recon means it is the smallest unit of the recombination. Okay, so the right answer of the cistern is genetic unit of the biochemical function, and it is nothing but gene. And winged bean is the popular name of the, so that is Sophocarpus tetragonolobus. So this is the right answer of this winged bean. Okay, Prosocarpus tetragonolobus. The feature unique to monocotyledonous root is, my dear friends, remember, so monocotyledon roots are polyarch condition. Okay, polyarch condition. If it is a dicot root, it is a triarch or tetrarch condition. Okay, triarch or tetrarch. But it is a monocotyledonous root. The vascular bundle the vascular bundle are we can called as polyarch xylem. Okay. Then the part B, 25th question. Which of the following statement about keratin protein of hair, wool, and nail is false? My dear friends, remember, keratin protein of hair, wool, and nail, it is a non-fibrous structural protein. Okay, it is a non-fibrous structural protein. It is present in the exoskeleton part of the living organism. Okay, nails, hairs, okay, and wool. Then the charcoal rot, also known as dry weather wilt, is caused by, that is Macrophomia phaseolina. Macrophomia phaseolina is caused of charcoal rot, also known as dry weather wilt is. Okay, wilt. Then electroporation is an experimental technique adopted in, it is especially biotechnology, genetic engineering. Okay, biotechnology, genetic engineering most abundantly used this electroporation process for the separation of the DNA bands or DNA fragments, DNA bands. Okay, then 2,4-dichlorophenoxyestic acid, 2,4-D, it is a synthetic oxide which is used for the herbicide. It is a weedicide or herbicide in the dicotyledon crops, for the monocotyledon crops for the removal of the dicotyledonous broad-leaved weeds. Okay, so 2,4-D is not a pesticide, it is an herbicide. It is a herbicide. And C4 plants are different from the C3 plants. We know that number of ATP molecules that are consumed in preparing sugar, it is difference between C3 and C4 plants. Okay. Then the matching the following. So this is the rockweed. Rockweed we can call as fucus. Fucus we can call as rockweed. And gulfweed, sargassum we can call as gulfweed. Okay. And reindeer moss. reindeer moss, we can call as cladonia. Reindeer moss, cladonia. And the last one, Irish moss, that is Chondrus. So Chondrus crispus. Okay. Then, so this is Amphidiploidy has played a major role in the production of new species of Brassica. The chromosome number of Brassica gentiana, an amphidiploid that is produced by crossing Brassica nigra. So the chromosome number 2n, the diploid chromosome number is 16, with Bacillus compressed is the chromosome number, diploid chromosome number is 20. So the producing this the number is 36. Okay, the answer is 36. Then tick-tock complex is associated with the import of proteins into. So this is very, very important question. Tick-tock complex is associated with an important protein, import of proteins into chloroplast. Import into chloroplast. Then 34th question. Which of the following genera has two polar nuclei in its mature embryo sac? That is Orchis. Members of which of the following families usually lacks endosperm? Very, very important. So without the absence of the endosperm, we know that orchids, Orchidaceae family. So orchids seeds are very minute seeds in the entire plant kingdom. And even though the endosperm is absent in Orchidaceae family or orchids, okay, then which of the following class of genes does not act as transporters in plants? So here we know that pin proteins, AP2 ERF proteins, ABC transporters, and aquaporins. So among these four, so AP2 ERF is not acted as a transporters in the plants. So this is not the, so this option is not acted as a transporters. Then the right statement here. So statement 1, PS2 mostly occurs in the stacked regions of the thylakoid. Yes, we know that. And PS1 is predominantly present in the unstacked regions of the thylakoids. Yes, it is also right. So that's why, so both statements are correct. So PS2, photosystem 2, mostly occurs in the stacked regions of the thylakoids. It is also correct. And PS1, photosystem 1, is predominantly present in the unstacked regions of the thylakoids, which is also correct answer. So both statements are true among these four options. Then apomixis is the type of reproduction which results in. Remember, my dear friends, it is an asexual reproduction type, apomixis, without involvement of the gametes or without sexual reproduction. Apomixis we are observed. So in this question, so both the options are correct, my dear friends. Development of the plants without fusion of the gametes is also right answer. Development of embryo from tepal cells, okay, or tapetal cells of the what we can say that antheridium or anther. Okay, then pectin, a component of plant cell wall, is stained by. So pectin, we know that pectic substance is very abundant. We are observed in the lower plants like blue-green algae and other aquatic or hydrophytic plants or aquatic plants. So whenever the pectic substance is diluted or dissolved in water, the thallus looks like a slimy nature. So whenever we are going to touch, that is very slippery in nature we are observed. So this pectic component of plant cell wall is stained by ruthenium red. Ruthenium red. It is very, very important question, my dear friends. So ruthenium red is used for the staining of these pectic components of the plant cell wall. Okay. Then the characteristics of curling of root hair. So curling of root hair, very, very important in the leguminaceae plants in the process of the nitrogen fixation by the biological nitrogen fixation. So it is very, very important in the assimilation of the nitrogen. Okay. Then which one of the match the acronyms? So listed here, the first column related to from the present second column. So this is CRISPR cross. So CRISPR, which is means clustered regulatory interspaced short palindromic repeats. CRISPR means CRISPR cross genes. We know this CRISPR cross technology. So this is advanced technology. The CRISPR, the abbreviation of the CRISPR is clustered regularly interspaced short palindromic repeats. And RNAi. RNA means ribonucleic acid interference or RNA interference. So then the third one is TALEN. So TALEN means transcription activator like effector. See here, effector nuclease. Okay, transcription activator like effector, not endo, effector nuclease. Then miRNA, that is now microRNA. miRNA means microRNA. The next, significance of Reynolds number is very, very important. Significance of Reynolds number is inertial force or viscous force. Reynolds number. Significance of the Reynolds number is inertial force, inertia force or viscous force. Then, if a single somatic cell in a multicellular organism harbors a mutation, it has no phenotypic effect. It has no phenotypic effect. Then alpha D plus glucose and beta D glucose are, that is anomers. Coenzyme, that is vitamin. We know that B2, some of the B2 vitamins and B complex vitamins, they are acted as the coenzymes. And competitive inhibitors, succinic dehydrogenase by malonic acid. Okay, malonic acid in Krebs cycle we are observed. Trypsin. So trypsin, that is alkaline pH. And Km and Vmax, that is, we know that Mendelian constant. Okay, and alpha helix. So alpha helix, it is a right-handed. Okay, right-handed. How many EDTA? Ethylene EDTA. Ethylene diamine tetraestic acid. Ethylene diamino tetraestic acid. So that is ethylene, EDTA abbreviation. So EDTA molecules are required to make an octahedral complex with calcium ion. How many calcium ions? So 1. How many EDTA molecules are required to make an octahedral complex with calcium ion? So 1 EDTA molecules are required. Only 1 EDTA. Ethylene diamine tetraestic acid, EDTA abbreviation. So that is also important. Then which one of the following is not correctly paired? See here, magnesium chlorophyll. So magnesium is the component in the formation of the porphyrin ring. So among that, magnesium is present in chlorophyll. And copper, it is a component in cytochrome oxidase. Okay, then molybdenum and ferrous. Along with molybdenum, ferrous is also a component in the nitrate reductase or nitrogenase enzyme or nitrate reductase enzyme. So here, manganese is not related to nitrogenase. So nitrogenase is related to molybdenum ferrous, but manganese is related to photolysis of the water or photooxidation of the water. So in photosynthesis, oxygen, water is split in the presence of the manganese ion. Then a student wanted to prepare 2500 milliliters buffer containing 100 millimole sodium acetate pH 4.0 containing 0.01% NaCl sodium chloride. He has the stocks of the 2 moles of the sodium acetate buffer pH 4.0 and 10% of the NaCl solution. What volumes of the given stock solution he has to mix in distilled water for preparing 2500 ml of the above reaction buffer. So that is the right answer is 125 milliliter of sodium acetate and 2.5 milliliter of NaCl. So this is the right answer. Then identify the mono unsaturated fatty acid given below. So that is oleic acid. Oleic acid is mono unsaturated fatty acid. So unsaturated fatty acids are very important for our body. Then, so match the cell organelle in group 1 with functional listed. You know that peroxisome, it is used for the detoxification. Mitochondria, we know that proton gradient formation for the ATP synthesis in respiration or cell respiration, aerobic respiration. Ribosomes also are used for the making of the proteins or protein synthesis. And leucoplast, we know that storage of the food material in the starch, like a starch granule. And the basis of precipitation, the basis of precipitation of proteins by ammonium sulfate is best described by which of the following statements? So the right answer is ammonium sulfate binds water molecules, making them less available for hydration of proteins. So this is the right answer of this question. And match the following from the disease with pathogen. So which disease caused by which pathogen? See here. So number 1, Creutzfeldt-Jakob. Very, very important, my dear friends. Creutzfeldt-Jakob is the disease caused of the prion. So prion is caused the mad cow disease. Mad cow disease in cows. Whenever the human beings, whenever they are eat the mad cow flesh, the person affected this Creutzfeldt-Jakob disease. So Creutzfeldt-Jakob, it is a disease whenever the human beings or persons who will going to eat the flesh of the mad cow diseased flesh. So that is Creutzfeldt-Jakob. It's caused by prion. Prion is only ability to infect the protein part of the virus. So protein part of the virus we can call as naked protein, we can call as prion in virus. Virus part. Prion is the virus part without presence of the nucleic acid. Then pseumocysts. So pseumocysts caused by fungi, and Legionnaires disease caused by bacteria, and rabies caused by virus. And one student from Kozhikode, that is Kerala district, got his admission in Wuhan University, that is China. So for PhD, he traveled to Wuhan under university during first week of January 2020 to start his semester. Within 1 week of his arrival in Wuhan, he became sick and consulted University Health Center. Based upon his symptoms, doctor immediately suggested him for viral test. So his viral test report was positive. Which disease pathogen was caused by suffering from? So this is simple. So the person who affected by typhoid disease, so typhoid is identified by this viral test. So typhoid is caused by Salmonella typhimurium bacteria. So this Salmonella typhimurium bacteria pathogen caused of this typhoid disease. So whenever the person suffering from this typhoid, we can use the test, viral test. Okay. So one scientist has given genomic DNA sample of a plant species to his research scholar, asked him to amplify a specific gene of interest XYZ. So that is whatever it may be. Using the available components in the lab. As suggested, he mixed all the components and performed the experiment, which was finally failed. Upon investigation, his supervisor realized that his student forgot add one very important component to the tube. His student mixed the following in the tube: genomic DNA, yes, okay. Then pair of the primers, okay, and 4 types of the DNTPs, okay, and buffer and DH2O. Okay. And which one is the forgot? Gurtupetukondi. My dear friends, remember. So talc polymerase is one of the most important enzyme for the control over the or sustain for the high temperature. So talc polymerase, without talc polymerase enzyme, so this genomic DNA experiment or DNA fingerprint test is impossible, or they failed for the correct test or correct identification. Okay, talc polymerase is most important. So genomic DNA important, pairs of the primers, 4 types of the DNTPs, and buffer and DH2O along with talc polymerase is most important. So it is nothing but DNA fingerprinting test. So whenever the thieves or any some of the parents and baby problems we are observed in the so they are going to DNA test from the forensic laboratory. So they are using this type of test. So along with these components, talc polymerase is most important. And which of the chemical formula belongs to valeric acid? So valeric acid formula is CH3, CH2, 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 COOH. It is the valeric acid formula. Okay. Then what is the term amphipathic means? Amphipathic means a chemical compound or biomolecule which possesses both hydrophilic and hydrophobic properties. So both the properties in the same compound. So that nature we can call as amphipathic. Okay. Amphipathic means hydrophilic nature means to attract. So this is invite or allowing the water, or hydrophobic, that is distracting of the water molecule. So this both the natures present in the same compound that we can call as amphipathic. Okay. Then when a protein solution containing tyrosine and tryptophan, so these are both the amino acids, warmed with concentrated nitric acid has turned yellow color because of nitration of aromatic ring. This test is called, that is xanthophoretic test. What test? Xanthophoretic test. Okay now. So this is about the xanthophoretic test. Then when diazonium fluoroborate was dried and heated, it gave aryl fluoride. This process of preparing fluorobenzene is called, that is Szyman reaction. Szyman reaction. Which of the following reaction is popularly known as Witting reaction? So Witting reaction means treatment of aldehyde and ketone with phosphorus to form alkenes, that we can call as Witting reaction. Then which of the following human disease is caused by helminth? So that is trichuriasis disease caused by helminthes. Okay, trichuriasis. So when ethylene is treated with sulfur monochloride, a toxic product is obtained. This product is mustard gas. Then in plant biology studies, some of the abbreviated here and used with a listed column A and column B, which of the correct one? So XA21. So XA21, it is a rice disease resistant gene. Okay now. Then SMAV1 related to a restriction endonuclease enzyme. And DAP1 or DAPI, that is a type of fluorescent dye. And RFLP, it is DNA based marker technique. AFLP, RFLP. So these are the marker techniques, DNA based marker techniques, RFLP, DNA based marker techniques. Then which of the following is considered as a secondary metabolite of higher plants? So secondary metabolites, carotenoids, is also considered as secondary metabolites in all higher plants. And morphine was initially isolated from Papaver somniferum. Papaver somniferum, unripened fruits. Unripened fruit of the Papaver somniferum, morphine was isolated. Okay. Then Ocimum tenuiflorum belongs to Lamiaceae family. In earlier Labiatae, we can call as Labiatae, but now we can call as Lamiaceae family. And plant tannins are, so tannins are polyphenolic components. So the tannins are used for the cleaning of the leathers. So leather industry, majority of the leather industry, they are depends upon the tannins for the sponge nature, for the making of the smooth nature of the leather. Then, so here common name and scientific name: finger millet, foxtail millet, pearl millet, and oat millet. Finger millet scientific name is Eleusine coracana. Foxtail millet scientific name Ceteria italica. And pearl millet scientific name Pennisetivum glaucum. Oat scientific name Avena sativa. Okay, so in Avena sativa, so the auxin experiments or phytohormone experiments done by F.W. Wendt. Okay. Then here some of the enzymes are here. So lysine for positive charge R group, aromatic amino acid, which is a tyrosine. Then cysteine contain sulfur, and arginine contain guanidino group, and alpha helical structure contain alanine. Okay, so these are amino acids, you know, lysine, alanine, tyrosine, cysteine, so arginine. These are all amino acids. So amino acids contain different groups, you know. Arginine contain guanidino group, cysteine contains sulfur, alpha helical structure contain alanine. Okay, alanine amino acid contain alpha helical structure. Then here Rho and RAC proteins. So Rho proteins or RAC proteins are GTP binding proteins, and RB tumor suppressor gene, and MAPcase serotonin threonine kinase, BAD apoptic factor, apoptic process or factor, BAD. Then inbred is nothing but cross-pollinated plant progeny. Pure line, that is self-pollinated plant progeny. Clone, vegetatively propagated plants. Hybrid, first filial progeny. Then in Escherichia coli 4 high frequency HFR strains donated the following genetic markers shown in the order donated. See here, these are the order: strain 1, strain 2, strain 3, strain 4. So all of the HFR strains are derived from the same F+ strain, that is a donor strain. What is the order of the markers on the circular chromosome of the original F+? So that is answer QWDMTPXACNBQ. So this is the order. This is the order of the original F+. Okay. Then miRNA, that is miRNA means microRNA. miRNA means microRNA, have been shown to play a significant role in gene expression. Some microRNAs induce gene silencing by binding to messenger RNA and inducing inhibition of translation. On the other hand, there are microRNAs that bind to messenger RNA and activate their degeneration. The following characteristic can be applicable to microRNA that inhibit mRNA translation. So that is the correct options are number 1 and 3. That is, microRNA is partially complementary to a region of target messenger RNA in the 3' UTR, and microRNA base pairs with messenger RNA through 6 to 7 nucleotides at its 5' end, referred to as seed sequence as well as few additional elsewhere. So 1 and 3 are the correct answer of this above question. And when the hybrid ratio 9:3:3:1, when it is gets modified 9:3:4 due to recessive epistatis. So 9:3:3:1 is the phenotypic ratio of the Mendel dihybrid cross. At the same time, the Mendel dihybrid cross ratio 9:3:1 phenotypic ratio has modified into 9:3:4. That we can call as recessive epistasis. Okay. And promoter is, what is the promoter? Promoter is upstream DNA sequence of a gene which is recognized by RNA polymerase in order to initiate transcription. And 75 question regarding the relationship between organism and ecosystem. See here, commensalism, one organism benefits but other is unaffected. Mutualism, both organisms are benefited. Mutualism means that is nothing but symbiosis, you know. So mutualism, both the organisms are benefited in the symbiosis. Commensalism is only one organism benefited, other is not affected. In parasitism, one benefited, the another one is definitely harmed. Amensalism means one impending the success of the other. One impending the success of the other, we can call it as amensalism. Then mutation is in a gene X and Arabidopsis thaliana results in more number of lateral root formation. Which one of the following correct answers? See here, my dear friends, the gene product act as a positive regulator of the lateral root formation. Simply, okay. So lateral root formation by the involvement of the pericycle of the vascular bundle, or even though the in the inner side of the stem or root, okay, especially root, so lateral roots. Then identify the correct corresponding target sites. See here, ECO-R1, it is a restricted enzyme which is target site is ECO-R1, target site is GAATTC, very, very important, my dear friends. And BOMH1, so that is GGATCC. HIND3, that is AAGCTT. Not I, that is GCGGCCGC. So this is the target site of the DNA fragment or DNA by the identification of the restriction enzymes or target site. And the scientists H.J. Muller had discovered mutagenic effect of X-rays in Drosophila melanogaster flies and CB bridges or C bridges. So bridges, non-disjunction proof of chromosomes containing genes. And Barbara McClintock is the discovery of mobile genetic genes elements, which is also called jumping genes. Okay, so which is also called as jumping genes or mobile genetic elements discovered by Barbara McClintock in GeoMaze. Okay, and Thomas Hunt Morgan was called as a father of modern genetics, and he discovered sex linkage. Okay, then see here, biosensor. So can be form a part of the biosensor. Enzyme, antibody, lipid, vitamin, both all are can form as a part of the biosensor. So all are right answer. So all are correct. Then match the aneuploid condition of a diploid organism given a panel. See here, nullisomy. Nullisomy means, so we can call as 2n-2. So nullisomy 2n-2. So loss of both members of the homologous pairs of the chromosome. And monosomy. Monosomy means 2n-1 means loss of a single chromosome. Trisomy 2n+1 means gain of a single chromosome. Tetrasomy means 2n+2. That means gain of the 2 homologous chromosomes. Okay, then so here the sequence we can DNA primer here, the backward primer pair of the below given gene. So this is the 5' and this is the 3' end of the DNA, and this is the forward prime, this is the backward prime. So this is the right answer of this above question. Okay. Then home box sequence is conversed. So conserved sequences which regulated development of many animal species. See here, International Crop Research Institute for the Semi-Arid Tropics, that is nothing but CUCET. So CUCET situated in Hyderabad, CUCET, Patan Cheru. CUCET situated at Patan Cheru, Hyderabad. Then National Institute of Virology, NIV, situated at Pune. Indian Institute of Horticultural Research. Okay, so it is situated at Bangalore. Indian Institute of Forest Genetic and Tree Breeding. So that is Coimbatore, Chennai. Okay. Then ribulose 1,5 bisphosphate carboxylase. It is an abundant enzyme in the entire plant world. That is also we can call as rubisco. So rubisco is also we can call as carboxydimutase. Very, very important. Carboxydimutase. Rubisco, ribulose 1,5 bisphosphate carboxylase. Oxygenase is also called as carboxydimutase. So which of the following are involved in the derivation of induced pluripotent stem cells of somatic cells? So that is SOX2, C-MyC, OCT4, KLF4. And the nitrogen fixation is not related to carried out by higher plants. So nitrogen fixation only prokaryotic living organism can ability to fixation of the nitrogen from the atmosphere. Why? Because nitrogenase enzyme and NIF genes. So the entire mechanism present in prokaryotic living organism like cyanobacteria, blue-green algae and bacteria. Okay. So that's why higher plants are not fixation of the nitrogen from the atmosphere. In Mendel experiments, the spherical seed character SS is completely dominant over the dented seed characters SS, small ss. If the characteristics of the higher height were incompletely dominant, such that capital T are tall and capital T small t are intermediate and small t are short, what would be the phenotypic resulting from crossing a spherical seeded short, that is capital S and small t plant to dented seeded tall, that is smallest capital T plant. So the right answer is all the progeny would be spherical seeded and intermediate height of this question. Then which of the following is not fairly correct? See here, niacin. So niacin deficiency, pellagra disease we are observed. Vitamin C deficiency, scurvy disease we are observed. Vitamin B12, pernicious anemia we are observed, Okay. Then beriberi is the deficiency of the vitamin B1. Okay. B1 vitamin deficiency, beriberi we are observed. Okay. Then maize seeds become viviparous when they are deficient. See here, abscisic acid. So abscisic acid, if absent in maize seeds, then automatically we are observed viviparous. They are viviparous. Which of the following is not present in plant cells? Centriole. Centrioles only present in animal cells. So whenever the plant cell is going to cell division, so the centrioles is produced from the cytoplasm of the plant cells. But centriole or centrosome is not prepared, but the tubules, so microtubules are made of the spindle fibers. Okay, so that's why plant cell is not present, this centriole. And glycolysis, we know that glycolysis is one of the gateway of the all respiratory whether anaerobic respiration or aerobic respiration, glycolysis. So glycolysis consists of 10 stages, 10 stages, 10 stages which we are observed in the glycolysis. In this glycolysis, the correct statement is, so number 1, glucose 6-phosphate and glyceraldehyde 3-phosphate, then 2-phosphoglycerate. So these 3 are related to this glycolysis. Acetyl coenzyme A is formed or produced after pyruvic acid oxidative decarboxylation. So that's why 1, 3, 4 are the correct answer of this above question. Means glucose 6-phosphate, glyceraldehyde 3-phosphate and 2-phosphoglycerate. Then bulbosum method has been used for in barley to generate, that is double haploids. So bulbosum method is used for the generation of the double haploids in barley plants. So in this cryptochrome is one of the photoreceptor in the camophore or chromophore. Okay, cryptochrome. Auxins, this is inhibit the outgrowth of the auxiliary buds, but it is promote the apical bud development. Cytokinins prevention of the senescence. Gibberellic acid breaking of the dormants of the seeds and germination of the seeds also. Gibberellin and ethylene promotes senescence of the flowers and fruit ripening. Then which enzyme is involved in epigenetic inheritance? Epigenetic inheritance. So epigenetic inheritance caused by histone methyl transferase. Histone methyl transferase enzyme involved epigenetic inheritance. Then flower represent a complex array of functionally specialized structures that differ substantially from the vegetative plant body in form and cell types. Following are statements made regarding floral meristems. So the right answer is 1, 2, 4. Okay. Then the zDNA is one of the left-handed DNA. zDNA is left-handed. bDNA is right-handed, but zDNA is left-handed. Then 98, xerophytes. The plants grow in the place with scanty of the water. Hydrophytes, plants grow in water, may rooted in the mud. Also, mesophytes, that is the temperate moisture, the plants that grow under average conditions of the temperature and moisture. Halophytes, the plants which they are adapted saline soil. Okay, halophytes. Then the diploid chromosome number of geomes, that is 2n is equal to 20. The number of chromosomes of DNA molecules that are found per cell when this original cell progeny progress through the G2 phase of the mitosis, that is 20 and 40. 20 and 40. Why? Because DNA G2 phase, the entire chromosome number is diploid or doubling. So 20 means G2 phase, the chromosome number is 40. Then the cluster of the oxygen evolving complex of photosystem 2. Remember, my dear friends, oxygen evolving complex OEC is only present in the photosystem 2. Photosystem 1 does not contain this OEC, oxygen evolving complex. So the oxygen evolving complex which is present or contain manganese, oxygen and calcium. Okay, Mn4, O5, calcium. So this is the OEC complex of the photosystem 2. So this entire question paper related to 2020 CUCET common entrance test of the Central University Plant Sciences Botany PG entrance examination, my dear friends. If you have any doubt, please ask me. My phone number is 9400933848. So this is my WhatsApp number. If you have any doubt, please contact me. I will clarify your doubts. Okay. I will stop here, my dear friends. I wish you good luck.